Well, good afternoon and welcome to everyone that's joined the first of a series of webinars that the Empire Discovery Institute and the Biomedical Research and Discovery Department at the University of Rochester is co-hosting. Thanks so much to Molly Mesko and to Kate Jordan, who have really done a lot of work to help organize this, to corral the presentations and participants over the course of 5 webinars that we plan today and then later in the fall. It's my pleasure to welcome everyone. I'm Ron Newbold. I'm the interim CEO and Chief Business Officer for the Empire Discovery Institute. EDI, as we refer to it, is an accelerator or a venture studio that invests in novel early-stage discoveries from academic labs at the University of Rochester, University of Buffalo, Roswell Park Cancer Center, and increasingly with other institutions outside of our home institutions to identify promising technologies, invest in them to bring them to a point in which they've been, the targets have been validated, hopefully molecules have been identified that affect the target of interest, and we can begin to have conversations with biotech, pharmaceutical companies, or others about bringing them into their pipelines. It's my pleasure to introduce Martin Graham and Ted Torfey. I think you'll be incredibly impressed with their experience and our other presenters that you'll meet later in the fall. Martin is the former CEO of EDI, who's my predecessor here. He has a background in anti-cancer drug development and an amazing track record of developing programs across a range of conditions, has extensive pharmaceutical experience at places like Sterling Winthrop, Sanofi, J&J, has started 2 companies of his own, and I think you'll be incredibly impressed with his background and expertise. Ted Torfey, who will be our second presenter today, tag teaming with Martin, is the chairman of our scientific advisory board. Similarly, Ted has a background and expertise in toxicology and pharmacology, spent many years in the pharma industry at places like GlaxoSmithKline, at J&J. He was formerly corporate officer of the Office of Science and Technology at J&J and sits on a number of advisory boards and panels today. And as I mentioned, is the chairman of our scientific advisory board. So with that quick introduction, Martin, I'll turn it over to you and ask that you kick off the webinar, and welcome to everyone that's joined.
Great, thank you very much, Ron. So today, as Ron said, Ted and I will be presenting the first of a 5-part webinar series on the entire drug discovery and development process. And we'll be really kind of covering at a high level in this introductory webinar the process of drug discovery and development from the very earliest inception, typically that you see within an academic institution, where we have a novel new discovery, and then how to exploit these novel targets and pathways to develop drugs for unmet need that have great potential, both commercially and for potential new therapeutic medicines in the future. Ted's going to be really focusing on the initial stages today, the preclinical drug discovery and development process. And then I'll be in the second half of the webinar discussing the clinical aspects of drug discovery and development, all the way through to taking a product to registration with the FDA. We'll also be touching on some of the more strategic aspects of drug development, introducing some of the upcoming speakers and their topics that will go into each of the things that we introduced today in more depth, and also touching on some of the business development and commercial aspects that are important throughout this entire process. So with that, we'll kind of go into the first part of the discussion here, and as a little segue into the main part of the webinar, EDI, as Ron described, really sits at the juncture between academic institutions and the research that goes on at very early stages in the process, and the activities that happen downstream within the pharmaceutical and biotech industry. And there's often it's seen as a real gap between those two worlds, and that's for a number of reasons. It can be lack of funding. These are typically capital intensive activities, and also pharmaceutical industry knowledge that is typically not found or part of academic institutions. So EDI was created to bridge those two worlds and facilitate the development and commercialization of novel discoveries within academia and translate them to a point where they can be matured and picked up by biotech and pharmaceutical industry, ultimately to take downstream to the full development process to an IND and an approved product. So EDI is as Ron highlighted, is really a collection of pharmaceutical industry experts drawn from various disciplines. There's about a 15-member scientific advisory board, and we've picked a number of those SAB members to be part of this webinar series to give much more in-depth review and discussion of the various aspects of drug discovery and development. This is how we're planning on structuring the webinar series. So today we have the introductory session on drug discovery and development, the high-level overview that Ted and I will present. And then that will be followed by 4 additional sessions coming up in the fall. The first one on drug discovery from basic research to a potential therapeutic. We'll be really focusing on the drug discovery aspects and some of the key activities and strategies that are employed by organizations like EDI, but more widely within the biotech and pharmaceutical industry. The second session will be a translational research and development webinar from lab to clinic. And this will really primarily focus on small molecule drug development, as well as an emphasis on the importance of developing biomarkers early in the process to help guide drug discovery and development. And we'll get into that more as we go through the presentations. The third session will then switch from small molecule development into the development of biologics, specifically aspects of antibody, protein, peptide, cell, and gene-based therapies, and the different approaches that have to be applied to the development of this type of therapeutic approach. And finally, we'll be giving a session on commercialization with a particular emphasis on the importance of protecting these discoveries, the IP and patent positions that have to be secured around each of these programs, as well as guidance on how programs in the early stage can be funded, as well as how to seek and interact with potential partners within the biotech and pharmaceutical industry. All this information, all the links to the presentations can be downloaded from the EDI website. And as Ron and Molly mentioned, this webinar series will be recorded for future reference. So with that, I'll hand over to Ted, and we'll start to go through now the first part of the discussion around the early drug discovery and development process.
Well, thank you, Martin. I appreciate the introduction, and I'd also like to say that I appreciate the participation of the panelists and all of the attendees. This is always fun, and we tend to get a lot of very interesting questions. The first thing that I'd like to discuss is what makes an attractive drug discovery program? What excites an investor, and particularly early stage investors as EDI is? What interests them? How do they evaluate an opportunity? And what I'll do is go through what EDI does. But I should point out that this is pretty generic, and it will fit for just about any opportunity that is either emerging from academia or those that come out of a biotechnology company or a pharma company. First is the investigator. Their academic record, their grant funding, is their lab well supported, communication skills, presentation skills, their enthusiasm. Are they excited about the opportunity to create a drug? Do they want to learn about what it takes to make a drug? Highly complex, long-term, and expensive process. The second thing is the novelty of the opportunity, and I'll talk more about that. What makes it competitive, or what allows an opportunity to beat the competition, and how is it supported with intellectual property? And when we speak of intellectual property, we are primarily talking about patents. There are other approaches as well, trademark and trade secrets. We won't talk a whole lot about that, but we're looking for the differentiation from the competition. Is there IP? Is there intellectual property? Has a patent application been filed? Or is there a clear opportunity to generate IP early in the drug discovery process? We also look at the degree of risk in that opportunity and what the mitigation strategy might be to either reduce the risk or to fail quickly if you're going to fail. And that's a theme that you're going to hear multiple times throughout the presentation. Unmet need and the market opportunity. Now, I'm going to speak from my own bias standpoint, but particularly as it applies to the nature of what EDI does and the early stage opportunities as an independent organization that we can help evolve and help with creating startup companies. The most important thing is the unmet medical need. Is there a gap in therapeutics or in treatment that can be addressed with the approach that is being taken? Market opportunity is somewhat less important in terms of the size. And the reason I say that is that if you're an investigator in a pharma company, if you can't tell them it's one or two, if you can't assess it at a 1 or 2 minimal billion opportunity, it's probably not going to fly. Well, the difference with a startup, an independent startup company, is that the entire pharmaceutical and biotech world is your potential customer. That means that you don't have to have a huge opportunity. You can have a niche opportunity or an orphan opportunity that can be just fine if you find the right company to partner with in the later stages or the latter stages of development. We also assess on the likelihood of follow-on investment. We would hate to invest in any early stage investor or angel investor, would hate to invest in an opportunity that succeeds to the extent that they can take it, or to the distance through the pipeline that they can take it, but then falls off a cliff because there aren't going to be investors who are interested in making larger investments at that point to bring the opportunity to the market. Target validation and the program stage. I'll talk more about this in the following slides. The target validation simply links modulation of the molecular target that you're expecting, or the expected modulation of the molecular target, activation or inhibition, that it will have an impact on the pathophysiology of the disease. How strong is that linkage? And the program stage. As you will see in the following slides, EDI will begin their investment only once an opportunity has reached a certain stage in the discovery continuum. And then finally, research plan. The focus of the research plan should be on drug discovery, not off into tangents, with well-described milestones and objectives. It should be technically feasible to do. Risks and the mitigation plan with clearly marked milestones and go, no-go checkpoints. And then, as Martin just alluded to, the strength of the biomarker and the target engagement opportunities. Do they exist? And are they rather, are they technically feasible to achieve in a reasonable amount of time? So moving on to the next slide, what do we consider in a differentiated product? What are we looking for? As I pointed out, the first and foremost criterion is, will the product that is envisioned address an unmet medical need? On the other hand, or in addition to that, will the product deliver economic benefit to the patient, to the employers, to society, to the healthcare system at large? Will it reduce overall medical costs for the individual who is going to be taking the therapy? Does the product have an improved safety profile over the competitors? Doesn't necessarily have to be first in class, but you have to have a pretty clear pathway to being the best in class. Does it have broader applicability for more conditions than the competitors? Can it be used for multiple indications? Can the drug be offered at a substantially lower cost to the patients or the payers? In general, for example, large molecules or biotherapeutics, as I will discuss in a moment, are much more expensive than small molecules. Can a biotherapeutic be replaced with a small molecule? Or can multiple drugs be replaced with a single drug? Will it improve the patient experience? Dosing and improved dosing frequency or route, fewer side effects, better efficacy, etc. And does the product have, or could the product have, a greater patent or market exclusivity than the other drugs that are currently on the market or coming? Usually it's coming to the market because that's what you're looking at for the competition. They're not necessarily on the market yet. But I will say that this one really only counts if you have one or more of the other attributes that are shown above. I should point out that a novel mechanism of action alone is not sufficient. A novel mechanism only counts if it can then lead to one or more of these attributes. All of these will lead into crafting right at the outset of a drug discovery program the target product profile. And this is the characteristics and the beneficial attributes that you expect your therapy to have. This is a target product profile which gets filled out and modified as you learn more about the targets and more about the therapeutics that you can use against the target. This target product profile ultimately will lead into, as Martin will talk about, will lead into the label that the FDA will allow you to have. The target product profile is what you hope to get. The label is what you do get and what you can market the drug on. I won't go through this in any kind of detail. I just wanted to make a few points. There are options, multiple different options in terms of therapeutic modalities or therapeutic platforms that can now be applied to the target that you have in mind. When I joined the industry, the pharmacopoeia had only small molecules in it. And these were joined with eye of Newton, toe of frog. It was that long ago, toe of frog. It was that long ago. The increasing complexity, as you go down this list, the complexity becomes greater. These are also newer modalities. So not only does the complexity and the size, essentially, of the therapy get larger, the regulatory pathway is less assured. It's less clear. And in fact, today, that regulatory pathway continues to evolve. Now, you'll hear about all of these throughout the sessions that we will have throughout the next 4 sessions, and you'll hear about them in detail. Today, we're going to focus on small molecule development and discovery, which serves as a very good basis for comparing the discovery and the development of these other therapies. We'll also say a few words about biotherapeutics, antibodies, enzymes, replacement factors, and those sorts of therapies. So in a linear fashion, these are the stages of drug discovery and drug development. Preclinical drug discovery is shown on top, and that's what I will go through with you. Clinical development is shown on the bottom. To point out that from the outset of target identification and validation through an NDA approval, we're looking at somewhere between 12 to 17 years. And you've also heard the cost, and both I and Martin will talk more about this. You've heard about the cost of putting a new pharmaceutical on the market, and it's somewhere between $1 and $3 billion with the associated risk, which we will talk about. So let's move on to the small molecule development pathway, or the small molecule discovery pathway. The value creating milestones and the checkpoints. There are 4 value creating milestones. It's at the interface between target identification and validation and identifying a hit. And I'll talk more about what that hit might look like and the characteristics that it has. So it's target identification and validation, a pharmaceutically acceptable lead, and I'll describe what that is, the selection of a development candidate that will go into investigational new drug or IND enabling studies, and then the IND submission to the FDA. The earliest entry point for EDI is typically on a small molecule effort is that you have identified one or more hits. One or more hits have been identified, and these hits are now ready to be modified into lead molecules. The approximate time frame is shown on the bottom for each of these, and we're talking about something of up to 6 to 6.5 years. And the overall success rate from target identification to IND submission in small molecule small molecule drug discovery is somewhere between 10 and 20%. If your career goal is to get a new drug, a novel entity, a novel chemical entity into humans to test a clinical hypothesis, most of us are going to fail. It's not because we did anything wrong. It's just because the inherent risks are so high. Just a quick comparison of biotherapeutics and small molecules. Shown on the top, biotherapeutics and small molecules, which I just showed you, are shown on the bottom. On the bottom, for small molecules, the time frame is 3.5 to 6 years with an investment of $15 to $22 million to have an open IND. Biotherapeutics are a little bit different, and this is really specifically for antibodies, although factors and enzymes fall pretty close to this. It's a shorter time frame. It's 2.5 to 3.5 years, and it costs less, about 8 to 10 million. Primarily because it's a shorter development time, or a shorter cycle time. It doesn't take as long to do it. Importantly, as shown on the top, the success rate is much greater. It's 30 to 50%. Sometimes it's even more than that. Well, why is that? Well, specifically when we're talking about monoclonal antibody discovery, once the target identification and validation is completed, once you have the antigen identified, the likelihood is that you're going to identify with multiple technologies at hand. You're going to identify a lead. You will be able to develop a cell line to express and to generate that antibody for clinical studies, and you will be able to manufacture the studies for IND enabling, manufacture the molecule for IND enabling studies. I'll also point out that it is, I won't say it's rare, but it's fairly unusual that an antibody falls out during that IND enabling study time frame. It's not, antibodies are not associated with the same tox issues, the same metabolism issues, the same drug-drug interaction issues, the same off-target issues that small molecules are. That also is why, partly why the cycle time is so much less. This is a table of some major differences between small and large molecules. I won't go through all of this. I'll just focus on the bottom 5. And again, all of you will have access to these slides. Small molecules have the advantage of multiple routes of administration. Biologics, and certainly antibodies, on the other hand, tend to be limited to the parenteral routes, IV or sub-Q. On the other hand, this is counterbalanced by the fact that small molecules typically are given once a day or more, whereas antibodies can be given once a week, once a month, sometimes even once every 2 to 4 months. So the dosing frequency is much less. And so the decision, or the benefit, or the benefit and the decision that the patient makes is, will I suffer through a sub-Q injection, which frankly isn't particularly onerous, once a week or once every 2 months, vis-a-vis taking a drug, a small molecule drug, once a day. Now, on the other hand, small molecule drugs are inexpensively manufactured and easily produced as generics once the patent runs out. Large molecules, biologics, are costly and complex. They're expensive to make. That's one of the reasons they cost so much more than small molecules. But fully generic versions are not possible because of the complexity. What is possible is what are described as biosimilar biotherapeutics. Small molecules are low cost, certainly to make, and lower cost to payers than are the biologics, which are high costs. Now I'd like to just go through some of these, not in great detail, because you will hear about each of these steps in more detail in one of the later presentations. But let's start with the target identification and validation. Now, although it's shown as a single chevron, and this is a linear process, well, no, it's not really a linear process. Target identification and really the validation, the target validation continues throughout the discovery continuum, throughout development, and even after the drug is on the market. We're still learning more about what interacting with that target does in terms of physiology and pathophysiology. It really never ends. So the objectives in this early stage for target identification is to identify a pathway or a factor that is important to the disease pathophysiology, then identify within that pathway a molecular target that can be modulated by one of those platforms in that prior slide. And then it's the demonstration, the strength of the demonstration of the relevance of that molecular target to the pathophysiology. That is the molecular target. How is HMG-CoA reductase, for example, how is that implicated in the generation of cholesterol? And then how is cholesterol involved in dyslipidemias, to use an old example? The methods that are used are human clinical or genetic data. And I will say that VCs and funders love these data. How much that love is actually warranted maybe is a little bit questionable, but they do really like to see genetic data and human clinical data. You have genomics, proteomics, metabolomics, disease models, knock-ins, knockouts, knockdowns, overexpression of the target, and hopefully either tool compounds or antibodies that you can then use to show the linkage of the target to the disease. What do you want at the end of this target identification and validation phase? You want the molecular target to be identified, to show that it is that either one or more of those therapeutic modalities would be an appropriate one to use to target the molecular target. Strong evidence that target modulation will indeed impact the disease process, and that certain key assays and models have been established. You then go on to convince somebody to fund a little bit of work, maybe it's done in your own lab, to look at, or a chemist, if it's coming out of a chemistry lab, and as if you're a biologist, believe me, chemists are your best friends. Buddy up to them at this stage. The objectives are to identify one or more pharmacophores that are amenable to an optimization program. By pharmacophore, I mean sort of a core molecule or heterocycle that can be modified to identify or to start a structure-activity relationship. And at this stage, you also want to identify tool compounds that you can then use in vitro and in vivo models to strengthen the proof of concept. The methods that are used are high throughput screening, computational chemistry, crystallography, and NMR, and intuition. The intuition and the experience of the chemist can be an in invaluable aid. Chemists don't think like other people think, and in this case, that's a really good thing. You can check in vitro and in vivo models that are in place, and selectivity screening should be in place at that point as well, just to see if those hits have some degree of selectivity vis-a-vis other molecular targets that are in the same class. Excuse me. As far as the hit characteristics are concerned, what is a hit? You have activity against the target, obviously, with a potency of less than or equal to 3 to 10 micromolar, a degree of selectivity against other targets of that class. The structure-activity relationship, or SAR, becomes evident. That is, that the chemist can begin to predict changes that are made in the hit that will either increase or decrease its potency or its selectivity. There is a pathway. You don't necessarily have to have patentability at this point, but there's a clear pathway to it. There's a clear opportunity for IP, and that pharmacophore is amenable to lead optimization. There's chemical space to explore that will improve potency, selectivity, and patentability. This also, I will point out, is when EDI would be interested in that opportunity. Then there's hit to lead. We need to expand and refine the hit series to truly establish a clear structure activity relationship. We're looking to improve by chemical means the potency and the selectivity, and begin to introduce drug-like properties, the appropriate solubility or the lipophilicity, cell or CNS penetrability or permeability. I'll point out that this is an iterative process. Chemists will design new molecules, synthesize them. The biologist will test them in vitro. SAR analysis will be created. And you may even at this point do some very quick, either in vivo or in vitro ADME studies, looking for example, metabolic issues with the drugs and with the lead and how they might be fixed. Again, it's an iterative process that feeds back for design of further opportunities or further therapeutics. For a lead, you would like to have the structure-activity relationship established. You're at sub-micromolar potency. You're selective at least 10 to 30-fold for the target vis-a-vis similar molecular entities. Active in vitro and in vivo. There are manageable ADME characteristics. It's amenable to further optimization, and to do that, you of course need to be synthetically accessible, needs to be a molecule that can be changed, and it's patentable. Once you're at that point, you then enter the lead optimization phase, and this can take quite a while. And the reason for that is that you're trying to improve the nature of the molecule such that you're pretty sure that it's going to succeed, as sure as you can be, that it will succeed in these IND-enabling studies. And you've gone from a nice tool compound to something that really looks like a drug. So you'll expand the SAR, strengthen the patent position. You will develop target engagement and biomarker strategies and the assays. Target engagement is are assays that would be done in vivo, hopefully even in volunteers in the clinic, that show that your drug is engaging the molecular target of interest. Biomarker strategies are sort of a follow-on from this, showing that you have strategies in mind to assess whether the drug, when it engages the target, is changing the nature of the pathway such that you will be affecting the pathophysiology. For cholesterol, that's pretty easy. It'd be the measurement of cholesterol, for example. But of course, that doesn't tell you that it's going to decrease cardiovascular events. And that's a whole other can of worms that I'm pleased will be in Martin's section. Not that example, but that's when it's done. And then to identify the development candidate. The development candidate is one that goes into IND enabling studies. You're looking for very high potency with KDs or KIs less than 10 nanomolar, selective, the mechanism of action is understood, et cetera, et cetera. All of these are improving your ability or improving the predictability of success in those IND enabling studies and then in the clinic itself. The one thing I will point out once again is intellectual property. By the time you're at this stage, you have to have this nailed down. Not only the protection of the molecules that you have, but a forward-looking strategy to further protect and expand the intellectual property estate that has been established. I'll point out one other thing. As important as the go criteria are at each of these stages, no-go is equally, if not more, more important. Early stage funders have to be, by their nature, highly capital efficient. And the killer experiments need to be done. If you've worked on target to hit for 30 months and you're getting nothing, it's time to stop. I even argue it's probably at 6 to 12 months if you've worked that long and you just don't see anything that the chemists believe can be moved further. It needs to stop, and you go back and conserve the capital for the next target, or until something happens that is unexpected that allows you to identify a hit. Finally, I won't say a lot about this slide. Martin will say more about it in just a moment. The IND enabling studies are the bridge going from drug discovery to drug development. These typically take 12 to 18 months. The target is 12 months, but it sometimes doesn't work out that way. I've only listed partially, this is only a partial list of all of the activities that need to be carried out. Many more are carried out as well. The 5 that are on top that are highlighted in green, these are actual studies, actual laboratory work being done that need to fulfill FDA requirements. This is when you are entering a highly regulated phase of the R&D process, and I would argue that the pharmaceutical and biotechnology industries are the most regulated in the world. Martin will talk a little bit about those touch points with the regulatory authorities in the US and in the EU. These are critical importance. These are critically important. So these are the studies on top that will be conducted. On the bottom are all of the documents that need to be prepared in order to submit to the FDA for their review. Well, this is the downside, maybe. It's a lot of work to do. It's a lot of work to get done within 12 months. The good thing is, once you've submitted the FDA, once you've submitted the investigational new drug application to the FDA, and they have acknowledged the receipt of it, they have 30 days to either get back to you with concerns or not get back to you at all. If they don't get back to you after 30 days, you can then dose your first patient. And with that, I will turn it back to Martin.
Great, thank you, Ted. Let me just pull up the slides here. As I'm doing this, I just want to also mention that there is an opportunity for all the participants and attendees to submit questions. There's a Q&A tab at the bottom in the Zoom presentation. So at the end of the webinar today, we'll try and compile some of those questions and answer as many that we can in the time remaining. Can everybody see those slides? So the next stage is
Not yet, Martin. We cannot see them.
You can't see that. Okay, let
No.
me. Sorry. Can you see them now, Ron?
No, not yet. Here they come.
So to move into the clinical development, as Ted highlighted, all the activities up to this point are preclinical activities, and in the final stage of the IND-enabling studies, these are all activities that are very highly regulated by FDA, and a clear roadmap is laid out on how to take a molecule into humans for the very first time. There's various different techniques that we'll go through in later the later sessions, but one here is allometric scaling, where data in animal studies can be used to extrapolate to establish a safe starting dose for human use, and that's one of the kind of key aspects of the preclinical work, is to establish that safe phase 1 starting dose. The preclinical work is often then done in 2 animal species, usually rodent and non-rodent, and that's usually done to identify the likely toxicities that the agent may manifest, and what needs to be measured in patients. An important activity in that preclinical work is to develop a prototype dosage form and formulation. This doesn't need to be the eventual commercial formulation, but it does need to be sufficient to be able to go into humans for the first time to evaluate the early safety in a patient population or a healthy volunteer population. This is also the time when biomarkers will be often developed if there's an opportunity there. And these could be prognostic biomarkers that could eventually be used to as a companion diagnostic, maybe to identify patient subsets to study unlikely responders and non-responders. What is often used are quantitative predictive biomarkers, and these can be very useful tools, particularly for demonstrating early proof of concept. We don't want to wait until late stage clinical development before we see if the drug is engaging the target and bringing about the pharmacological effects that we think it is. And that's going to be discussed in more detail in the session that would be presented by Christine de Buch on the advisory board. So now we've moved from preclinical development into the clinical development phase. And I'm going to go through each of these phases of clinical development and just highlight the key activities that are performed typically in each of these stages for a small molecule. As Ted mentioned earlier in his presentation, we establish early in the discovery and development process a target product profile. And these are really the kind of ideal characteristics of the molecule that we're seeking in a development candidate. But as we go through preclinical development and into clinical development, that really starts to then migrate from the ideal properties of the drug that we're trying to target into the characterization to then define the established properties of the drug that ultimately will go into the package insert and the approved labeling. This scheme also shows the key regulated steps that FDA have with companies as a compound and drug goes through the various stages of preclinical and clinical development, with the IND submission, a checkpoint at the end of phase 1, a checkpoint at the end of phase 2. And again, these are often used by the companies as go, no-go decisions if the results, if the safety and efficacy of the compound isn't panning out as planned. One of the key meetings late stage is the pre-NDA meeting, and this will then be a discussion or a series of discussions with the FDA to really define what they're looking for in the final submission package that will go to them for review and approval. And then there'll be either an IND or in the case of a biologics, a BLA, biologics licensing agreement submission package for FDA review and approval. So the first series of clinical studies are the phase 1 studies, and these are typically done in healthy volunteers, although an increasing trend now within the biotech and pharmaceutical industry is to, as early as possible, evaluate the drug, if possible, in patients. And it's possible in certain instances, particularly oncology, for example, to be able to do that evaluation. They were really primarily a safety study and pharmacokinetics, looking at drug exposure. But as companies want to get more rapid evaluation of a compound, there's often opportunities to do some early biomarker studies to see, as the doses escalated within that patient population, the engagement of the target and the evidence of pharmacological activity. We're often, as I say, using prototype dosage forms and formulations. These will kind of develop and evolve during clinical development in tandem with these activities. And what we're trying to do within the phase 1 setting is really establish a recommended phase 2 dose that can then go into the target patient population of interest. We're also, as I say, trying to embed often now biomarker strategies into these phase 1 studies, surrogate measures of response, looking for early dose response relationships, and opportunities to establish clinical proof of concept. If we pass the phase 1 hurdle, the phase 2 setting is really an exploration of the drug in the target patient population of interest. And companies will often use this phase 2 setting to also expand activity and evaluation of a compound in other potential indications or patient populations that may expand the potential labeling use of that drug in the future. So these are typically studies that are more non-blinded, not statistically powered clinical trials. We're usually doing a lot of PK and PD, pharmacokinetic, pharmacodynamic evaluations. They're looking at the relationships between drug exposure and drug response. And this can be very, very useful for then using in modeling and simulation to come up with some very fine-tuned and optimized dosing regimens to take forward to the more definitive clinical trials in phase 3. We're also looking at safety and exposure, PK. Clinical proof of concept is a key endpoint for these studies, and often go, no-go decisions are made at the end of phase 2 if a compound is either passing the hurdles or failing in terms of the efficacy and safety, the readouts. What we're looking to establish here then is the recommended phase 3 dose that then can be taken into the pivotal registrational trials for approval. The phase 2 area, and we'll go into this in more detail in the future session, is really the place where biomarkers come into the full utility, where we can establish clear dose-response relationships many times, use that to develop computer-based models to try alternative or optimized dosing regimens, and help build the case to establish clinical proof of concept. If it's a go and a compound advances to phase 3, this is then the setting where the drug is tested in definitive statistically powered trials for safety and efficacy. These are usually 2 independent, well-controlled trials. They're statistically powered, often blinded or controlled with crossover designs. We're collecting a lot of detailed safety information as well as efficacy information. And what we're doing here is establishing the dose, the recommended dose that ultimately will appear in the product label. We're also, in this setting, because of the number of patients involved, able to look and mine the data sets for how the drug may react in certain specific special patient populations. It may be elderly, it may be patients with impaired organ function or performance status. And again, these are analyses that are performed that ultimately can help provide useful labeling information, particularly around dose modification in patient populations, if that's warranted. So those are the first stages running up to the registrational studies, but there's a whole series of activities that run in parallel with the phase 1, phase 2, and phase 3 studies. And these are the clinical pharmacology studies, again, important studies that are required for product labeling. And some of these even go back into preclinical. For example, the in vitro drug-drug interaction studies for a small molecule are typically done in vitro to evaluate any potential metabolic drug-drug interaction potentials. And if there's flags there, then studies are performed in patients to see whether or not they are potentially clinically relevant. Other kinds of studies that are performed as part of the clinical pharmacology package are human radiolabel studies. So this is where a C14 label is embedded in the molecule, and it's really to characterize the metabolic fate of a small molecule, its routes of clearance, and its mass balance. Where does the drug go? How does it distribute into tissues? What are the major routes of clearance, either hepatic or renal? And that's important, again, further on, as the FDA want to understand how a drug is cleared and if there's any need for dose modification in patients with impaired organ function, or in patients that have different characteristics. Either it could be due to ethnic differences in drug metabolism, gender, age, and then other intrinsic and extrinsic factors, smoking status, for example. The post-approval area is also one that the FDA want companies to follow up on. And an example here of a lot of activity that goes on after a drug is approved by FDA is in the development of agents for pediatric use. And this is driven by some relatively recent legislation to mandate companies to develop pediatric products where applicable. And these activities are commitments that companies take on as part of the overall approval package. Phase 4 studies can also be conducted, and these are usually labeling expansions, maybe looking at new patient populations or indications. So these are very capital intensive activities, and as Ted alluded to, in the early stages, up to $20 million can be required for a relatively lean and mean biotech company developing a small molecule. So in the early stages, that can be acquired. That type of funding can be acquired through grants like SBIR funds. But as we get into the lead optimization and IND enabling studies, that really is very capital intensive and often requires venture capital investments into the programs all the way through until late stage clinical trials, phase 3, where often it's necessary to engage with a biotech or pharma partner. So looking at the raw costs here from that cover the entire drug discovery and development program, these can be in the order of $300 to $600 million. And this doesn't include cost of capital, the cost of capital and account for program failures, which are multiples of these figures, often expenses to pharmaceutical companies during the development process. It's also a time-consuming process. Again, this can take anywhere up between 12 and 17 years to get a compound through the various stages of development, through the preclinical stages and through the clinical stages to approval. So what are the available sources of capital for funding this type of program? And we'll be going through some of this in more detail in the final session around the business and commercial aspects of drug discovery and development. But in the early stages, the research in academic institutions, often funded by grants, R01 and foundations. As we move into early drug discovery with institutions like EDI, we have funding available to bridge the gap from academic funding into the early translational funding. SBIR, STTR awards are often good sources of funding in this stage of the drug development. But then as we advance forward into late stage preclinical development and certainly into clinical development, angel and more commonly venture capital is required to support these activities, as well as the strategic pharma investments. I think one of the pitfalls that that can happen to many academic investigators is that they want to try and go it alone, and with non-dilutive funding. Non-dilutive meaning that we're not bringing on board venture capital where there's a share of the revenues in the future. I think what the difficulty is there is that there's a lot of funding is needed, as you can see, to really advance these programs all the way through. But it's also, I think, the lost opportunity and cost of time that if an investigator doesn't have the level of funding required to take a program through rapidly, that there's a lost opportunity and that patent clock is ticking away, which can be very costly. So one of the things that we want to really emphasize as part of this series, and again, this will be discussed in more detail in the final session on the commercial and business aspects, are the intellectual property considerations that go hand in hand with this type of activity. And this is such an important area that we wanted to focus on just the the approach that should be encouraged in early stage programs to get good patent protection on an invention, on a program, and it's really the cornerstone of the whole development process. It's important to have a very good, strong IP position, and I think what we need to develop here is how your product is, how does it differentiate you from the competition? Will it give you a competitive advantage? How will your IP really compete in a space where there's a lot of other existing competing IP? And then what strategies you can try and implement to strengthen your patent position. And one of the strongest primary strategies is to get what they call novel composition of matter patents. These are patents that protect the chemical entity or chemical entity in the series of analogs in case of a small molecule program. Those are some of the key patents that need to be secured before publication of a lot of the research and development activities. Once it's published, if a patent position hasn't been established, then it's not going to be possible to secure that in the future, and there'll be a considerable loss of value there. There are other kind of strategies, though, that you can do to enhance your patent position, and there are methods of use patents. In some instances, there's an opportunity to patent drug-device combinations, and even companion diagnostics, and that's a particularly common strategy in the case of oncology agents, for example, where a companion diagnostic to identify specific mutations for targeted therapy can provide a robust patent position in tandem with the composition of matter patents that have been established for the main products. And we'll be talking about these more in the final session. And then, as a final slide here, Ted and I wanted to just open this up and really kind of go through some of these, what we feel to be important considerations for discussion on investigator company roles and relationships and startups. And I think organizations like EDI and other accelerators and early stage drug discovery and development organizations really recognize the scientific founders' knowledge and expertise, and their network are really vital in advancing the early stage technologies. And the academics know the science. I think what is important is that there's an openness to be able to recognize that additional expertise will need to be brought in for that into the program for that to be successful. Ted, I'll hand that over to you and let you maybe discuss some of these other points here.
Sure, I'd be happy to. Early stage funders invest in advancing technology. They don't fund laboratories. The funding is directed toward advancing the opportunity or the program either to the next value creating inflection point or to a no-go decision. And I'll come back to that. This is a difference. You may detect a difference between what early stage funders do and most VCs, as opposed to what a pharma company or a larger biotech company might do, in that they might help to fund a laboratory. Typically, that does not happen with the early stage funders. Don't feel that it's a slight if early stage investors want to replicate key studies outside of the laboratory, that is by a third party, outside of the innovator's laboratory. It really doesn't have anything to do with whether they trust the results or not. What they want to do is to see how robust the key studies were and how robust the models might be. So oftentimes, before anything is signed and before any agreement is made, they will fund one or more studies that would be done either in another academics laboratory or in a CRO, a contract research organization. Be open to alternative therapeutic indications. You might think that the opportunity and the target that you have identified is most important in congestive heart failure. Bringing other people to the table will bring other ideas to the table. Sometimes the ideas aren't necessarily strictly based upon science. And that is, in the case of congestive heart failure, it may be that the pathway that is involved is just as important in another indication, but that the proof of concept can be done very quickly. And it can be done very quickly with fewer patients and less expensively than it would be done in congestive heart failure. Also, they may suggest to reduce the cohorts, to refine the cohorts that are going to be tested by asking the question, or maybe making the statement, if it doesn't work in this subset of individuals, if it doesn't work here, it's not going to work in the broader in the broader field of the indications. One of the things that you will find that funders, early stage VCs, seed companies, accelerators, what they'll really look for is help from the academic founders to manage the university bureaucracy. We have no ability to do that. We have no ability to manage them, nor any ability to influence them. We oftentimes will need your help in trying to accelerate the pace of, say, a license or the filing of a patent, and so on. I think most of you will understand that delaying the publication of new results is sometimes necessary to protect intellectual property. We know that there are two competing needs or two competing goals that you have. One is you need to publish. The other is, and in fact, that's a large share of the reason that you are doing what you're doing. But in addition to that, if a publication is made too soon and a disclosure is made too soon, it may negate the value of the opportunity that you have identified. If you don't have patent protection, you won't get funded. Typically, academic founders will receive equity in the startup rather than a salary or a consulting fee. This may be different from what you would find if you partnered with a pharma company, for example. They may have the financial means to give you a consulting fee. This typically is simply not possible when you're dealing with these highly capital efficient and capital restricted early stage funders. And finally, I mentioned this a little bit earlier. An early kill experiment, looking at it from the funder side, an early kill experiment is as important and sometimes even more important than a go experiment. If there's a critical experiment that can be done that says this is not going to be feasible, or we're not going to achieve that target product profile or get anywhere near it that we thought we were going to be able to do, there's nothing wrong with running the kill experiment, stopping the work, and then preserving that capital to move on to the next opportunity.
Yeah, and I think that's particularly important, for early stage programs where there's limited capital, and that's why I think organizations like EDI are very keen on introducing biomarker strategies as early as possible into the program, as these can often give early quantitative measures of whether or not a project or program is likely to be successful or not. So we're reaching the end of the formal presentation here. If there are questions that the participants would like to submit through the Zoom link, we'd be happy to address any of those that we can in the remaining time here. This is just the series of upcoming sessions again, and we'll be posting the specific dates and times of these webinars in the near future. So with that, we'd like to kind of open it up to the question and answer session.
So Martin, I'll start. And Martin and Ted, thanks so much for the presentation, the overview, all the work that you put into this. The intent is really to give the highest level overview of the overall drug discovery process. And then, as you've said, we'll get into the specific details in each of these phases in the fall. I think we do have a tentative date for that first session in September. It'll be September 18th. But as Martin indicated, we'll make note of the dates and the registration links on the website. I also want to thank and acknowledge for those participating today. There's a number of faculty listening today and that will, I'm sure, listen to this in the future. We also have representation from our tech transfer colleagues at our partner institutions. They are a great source of information and sort of sounding board for any faculty member that's thinking about, hey, I think I have a drug discovery program. I have a novel discovery that I'd like to seek funding for. What do I do? I really highly recommend that you speak to your tech transfer colleagues, and of course, reach out to EDI as well. If we can be helpful, we're happy to do that. That's really the point of these sessions, is to provide information, to provide education, provide the benefit of our collective experience in developing drugs. So I hope people will take advantage of that. Martin, I guess just to put you a little bit on the spot, but just thinking about, since you've created companies, you've been successful in taking that particular route. We often see faculty members wanting to start a company. As you described, going it alone through non-dilutive funding, just grant funding, but ultimately they then look to raise venture capital or other money. Can you talk a little bit more about that? How were you able to be successful there when we often see faculty members create companies, but then really struggle to raise money or to advance that particular company, and your advice to faculty members thinking about that?
I mean, there are examples, where faculty members have very successfully formed companies and acquired funding and, attract pharmaceutical partnership and investment. So it's certainly possible. I think that the challenge is that it's really a very full-time commitment to, go into a company creation and do all the different aspects of drug discovery and development, not only from a scientific perspective, but also from the business and fundraising perspective. So in our experience, the most successful way to advance a program, rapidly to a point where it could be investable and there could be company creation, is for academic investigators to really establish a professional executive team around them, people who are experienced in the day-to-day operations of drug discovery and development, who have had regulatory experience. They know the regulatory environment with the FDA, how to navigate, that pathway, and know where the potential pitfalls are along the way there. And also investigators who can then surround themselves with people who are, professional, and have been able to successfully raise, large sums of capital and engage with the business aspects of of the company, particularly if they're bringing in venture capital investment. So, I think it's not impossible to do that, but I think if you've got a good idea and you want to get that to a point of getting it into patients and generating a potentially approvable products, that would be the way that we've seen most programs be successful.
Great, thank you. I'll also, I think, make the point that many times, I think Ted and Martin, you both sufficiently and I think importantly covered the aspect of not publishing too much too early, because that creates prior art against yourself if you want to file a patent later. If you have a discovery or an idea that you feel could be a program that could lead to a novel therapeutic that could be brought to market, those are important aspects to consider. It's not to say, I think, as Martin said, we certainly acknowledge and recognize the importance of publishing, but you want to cover your ideas through patenting first, and then think about the strategy of when's the appropriate time to do that. And oftentimes, it's keep the information confidential for quite a period of time. We at EDI certainly have invested in programs that started with no filed patents at all, but the intent is definitely to file patents, cover the novel composition of matter that hopefully will develop through the course of a program. And then, of course, we recognize the importance of publication to all faculty members. So I think those are important points that were raised today.
Yeah, and it really is, I think the patent position is really the kind of cornerstone of everything here. If you don't have a strong patent position or ability to be able to develop a strong patent position, then it's unlikely that that product or that program would garner interest from funders, and may never reach the opportunity to develop that into a useful therapeutic. So it's an very important consideration, and we understand it's a difficult balance there, between the need to publish, but also the need to be able to protect an idea so that you can really develop it to its full potential.
Well, I want to thank you both. I think this has been a really great opening discussion. There's one question that just came through from Steve Feldon. How important is the use of large animals in preclinical evaluation and testing? And are rodents sufficient? I can't see the rest of your question, Steve, but perhaps Martin or Ted, can you start to address that? How are rodents sufficient, or how far should someone try to take their animal studies and the work that they do in their labs?
Yeah, again, I think any in vivo model has to be, as far as possible, representative of the human situation. And not only that, the regulators do mandate that both a rodent and non-rodent species are part of the early drug development program, particularly from a kind of safety perspective. So I think it's if you can find and justify a model that will satisfy the biology and also satisfy what the regulators want to see in terms of toxicology studies being performed prior to human testing, you can try and make that case. And there are certain instances where that may be possible. But for most part, I think the regulators will require you to perform at some stage a non-rodent study, which could be dog, could be non-human primate, or other models that would be kind of directed again by the biology.
Mm-hmm. Yeah, and to go a step earlier than that, and if we're talking about preclinical models for efficacy, that's a very difficult question to answer. Models are sometimes predictive, they are sometimes not. Typically, when they are predictive, it's after the fact. You only know it if it's a novel mechanism. You only know that the model predicted what happened what's going to happen in humans after you get into humans. You know, we know that there are certain models that are good for NSAIDs, for example. If it works in that model, it's probably going to work in humans. There is no requirement. There's no requirement from the FDA that you need to show efficacy in an animal model. They like to see it, and it gives them a sort of, when you're doing the risk-benefit balance, well, it makes them feel pretty good about it. But if you've done if your preclinical development package is good, and you're going to dose patients or volunteers at the doses that were safe, and there's a margin of safety that came out of the preclinical program, they'll likely be okay with that. After being in the industry for the decades that I have, it's animal models to me became a little bit less of a go for efficacy than using as a pharmacokinetic, pharmacodynamic measure. Are you getting something? Are you seeing a signal at a dose that is relevant to what's going to occur in humans? It's also, animal models are also often used as a no-go. If it should work in that model, based upon everything that we know, both of the human pathophysiology and the animal model, if you have adequate plasma concentrations and it's not working, or it doesn't give you the result that you would expect, that might tell you it's time to think about taking a different direction.
Great. Thank you.
I think another kind of advancement that's emerged in recent years within the pharmaceutical and biotech industry is computer-based models. And I think they can be they may not completely eliminate the future need for animals, particularly in efficacy studies, but there's some very sophisticated physiologically based models now across multiple species, from mouse to rat to rabbit to dog to primate to human, where the physiology is scaled across all those species, and you can take an early data, or maybe just physicochemical data about the molecule, and plug it into some of these advanced simulation models, and get in some cases quite predictive estimates of drug exposure, drug metabolism. And that can be used, I think, to more efficiently guide future studies in higher animal species, and even predict what you may see in patients and certain patient populations.
Great. Well, again, I think I'll take the opportunity to wrap things up there. Martin, Ted, thanks so much for this great overview. I hope the participants that are joining today and those that will watch this webinar over the course of the summertime will look forward to our next session in September. And thanks again to Kate and Molly for all the background work and pulling this all together. I hope it's useful. And again, feel free to reach out to us at EDI if we can help provide any kind of information or guidance that may be helpful to any faculty researcher. So thanks very much, everyone. Enjoy your evening.
Thank you.
It's been a pleasure.
You
too.
Bye.
Bye-bye.